5.3 RNA world. The big question. Which molecule came first as the genetic material? Scientific consensus. RNA was likely the first genetic material. Evidence supporting RNA as the first genetic material. Feature explanation. Dual-role RNA could store genetic information and also act as a catalyst, ribozyme. Enzymatic activity. Some biochemical reactions are catalyzed by RNA, not proteins, e.g. RNA splicing. Simplicity. RNA is simpler than DNA and capable of self-replication in early life forms. Why RNA was replaced by DNA. Feature RNA-DNA. Catalytic yes, ribozyme activity no. Stability less stable due to 2' OH group more stable. Reactivity high-low. Strandedness single-stranded double-stranded, with repairability. Suitability for long-term storage. DNA evolved from RNA through chemical modifications to become more stable and reliable for long-term genetic storage. Key concept. RNA is believed to be the starting point of life, forming the core of early life systems before the evolution of proteins and DNA. Important points. The concept of RNA world explains the origin of life. RNA served as both genetic material and enzyme in primitive cells. Over time, due to instability of RNA, DNA took over the role of genetic storage, and proteins became the main catalysts. DNA's double-stranded structure allows error correction and repair mechanisms. Exam-based questions. 1 MCQ. Which of the following supports the RNA world hypothesis? RNA acts as a genetic material only in eukaryotes. B. RNA acts as both genetic material and catalyst. C. RNA is always double-stranded. D. RNA cannot replicate. Answer. B. RNA acts as both genetic material and catalyst. 2. Assertion. Reason. Assertion. DNA is more stable than RNA. Reason. DNA lacks 2' OH group and has a double-stranded structure. Answer. Both assertion and reason are true, and reason is the correct explanation. 5.4 Replication. What is DNA replication? DNA replication is the process by which DNA makes an identical copy of itself during cell division. Key historical insight. Watson and Crick, 1953. The scientists who proposed the double helix model of DNA also suggested a mechanism for how DNA could copy itself. They wrote, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. The basic idea: DNA consists of 2 strands. During replication, the 2 strands separate. Each strand acts as a template to build a new complementary strand. Result: 2 DNA molecules, each with 1 old parental strand and 1 new strand. This type of replication is called semi-conservative replication. Definition. Semi-conservative replication. A mechanism of DNA replication in which each new double-stranded DNA molecule consists of 1 old parental strand and 1 newly synthesized strand. Result of semi-conservative replication. Molecule strand 1 strand 2. DNA 1 parental new. DNA 2 parental new. Each daughter DNA molecule is half old, half new. Hence the name semi-conservative. Importance. Ensures genetic continuity from one cell generation to the next. Maintains accuracy due to base pairing rules. A with T, G with C. Exam-oriented questions. 1 MCQ. DNA replication is called semi-conservative because a. If the whole molecule is newly synthesized. B. Half of the strand is RNA. C. Each new DNA has 1 parental and 1 new strand. D. Both strands are completely new. Answer Z. 2. 1 mark question. Who first suggested the mechanism of DNA replication? Watson and Crick in 1953. 3 short answer. Define semi-conservative replication. A method of DNA replication where each new DNA molecule contains 1 original, parental, strand and one newly synthesized strand. 5.4.1 The experimental proof. Semi-conservative DNA replication. Key experiment by Messelson and Stahl, 1958. They provided the experimental proof that DNA replication is semi-conservative, meaning each daughter DNA molecule has one old strand and one newly synthesized strand. Organism used. Escherichia coli E. coli. A bacterium that divides every 20 minutes. Steps of the experiment. Step 1. Growing E. coli in heavy nitrogen. Superscript 1H4L. E. coli was grown for many generations in a medium with superscript 1NH4L, heavy nitrogen. This superscript 1N was incorporated into DNA bases. Result: all DNA became heavy DNA, denser due to superscript 1N. Note: superscript 1N is not radioactive but heavier than normal superscript 1. Can be separated from superscript 1N DNA using CSCL density gradient centrifugation. Step 2. Transfer to light nitrogen medium, superscript 1H4L. Bacteria were now shifted to a medium with normal superscript 1H4L. Samples were taken after each generation, 20, 40 minutes, etc. DNA analysis. CSCL gradient centrifugation. Generation DNA composition density band seen. 0 minutes all heavy, superscript 1N/superscript 1. Single heavy band. After first gen 20 minutes. All hybrid, superscript 1N/superscript 1, intermediate band. After second gen 40 minutes. 50% hybrid, 50% light, superscript 1N/superscript 1. 2 bands, intermediate and light. Conclusion. The first generation showed only hybrid DNA, proves that one strand of old DNA is retained. The second generation had half hybrid and half light DNA, confirms semi-conservative replication. Final proof. Each DNA molecule has one original, parental, strand and one newly synthesized strand. Key terms to remember. 15N DNA equals heavy. 14N DNA equals light. Hybrid DNA equals 1 15N strand plus 1 14N strand. SCL centrifugation helps separate DNA based on density. HOTS question. Question: why didn't the DNA show a mixture of 15N and 14N bands in the first generation? Because DNA replicates semi-conservatively. Each new molecule had one 15N strand and one new 14N strand, forming only hybrid molecules. If E. coli was allowed to grow for 80 minutes, the number of generations would be 80 minutes divided by 20 minutes/generation equals 4 generations. Let's calculate the proportion of hybrid, superscript 1N/superscript 1N, and light, superscript 1N/superscript 1N, DNA after 4 generations. Generation hybrid DNA, percent. Light DNA percent. 0 100%, heavy 0%. 1 100% 0%. 2 50% 50%. 3 25% 75%. 4 12.5% 87.5%. Final answer. After 80 minutes, 4 generations, the DNA will be 12.5% hybrid, superscript 1N/superscript 1. 87.5% light, superscript 1N/superscript 1. Reason. In semi-conservative replication, hybrid DNA remains unchanged in number. New strands are always synthesized using superscript 1N, so light DNA increases exponentially with each generation. Taylor's experiment, 1958. Semi-conservative DNA replication in eukaryotes. Objective. To demonstrate how chromosomes replicate in eukaryotic cells, whether it is conservative, dispersive, or semi-conservative, as had already been shown in prokaryotes by Messelson and Stahl. Experimental organism. Plant used, Vicia faba, broad bean. Reason. It has fewer and large chromosomes, making it ideal for autoradiographic studies. Radioactive marker used. Superscript 3H thymidine, tritiated thymidine. Thymidine is a precursor for DNA synthesis, not RNA. Hence it marks only DNA. Experimental steps. Labeling phase, first generation. Root tip cells were grown in medium containing superscript 3H thymidine. As DNA replicated during interphase, S phase, the radioactive thymidine was incorporated into new DNA strands. After one cell division, mitosis, both sister chromatids of each chromosome became radioactively labeled, since both strands had received a newly synthesized radioactive strand. Non-labeling phase, second generation. The root tips were then transferred to normal, non-radioactive thymidine medium. Cells were allowed to divide again. Only one chromatid of each chromosome was radioactive, and the other remained unlabeled. Detection method. Autoradiography was used. Cells were fixed, placed on a photographic film and developed. Radioactive DNA showed up as dark grains, allowing visual proof of label distribution. Conclusion. The results showed that in the second generation, only one of the chromatids in each chromosome was labeled. This directly supports the semi-conservative model, where one strand of the parent DNA is retained. The other is newly synthesized. Summary table. Generation growth medium result. Chromatid labeling. First radioactive, superscript 3H thymidine. Both chromatids labeled radioactive. Second non-radioactive, only one chromatid labeled in each pair. Why it was important. Proved semi-conservative replication not just in prokaryotes E. coli, but also in eukaryotes, plants. Provided visual proof using autoradiography. CQ practice. What radioactive compound did Taylor use in his experiment on Vicia faba? A. Superscript 3H uracil. B. Superscript 3H thymidine. C. Superscript 3 superscript 2p. D. Superscript 3 cytosine. Answer: B. Superscript 3H thymidine. Which model of DNA replication was confirmed by Taylor's experiment? A. Conservative. B. Semi-conservative. C. Dispersive. D. Progressive. Answer: B. Semi-conservative. In the second generation, why was only one chromatid radioactive? A. Both old strands were radioactive. B. DNA replication was dispersive. C. Only one new strand was synthesized. D. One old strand paired with a new unlabeled strand. Answer: D. One old strand paired with a new unlabeled strand. 5.5 Transcription. Definition. Transcription is the process by which genetic information from one strand of DNA is copied into RNA. Key concepts. Complementarity is the base pairing principle followed during transcription. Adenine A pairs with uracil U in RNA instead of thymine. Unlike DNA replication, only a segment of DNA is copied, not the whole molecule. Only one strand is used as a template. Why only one strand is transcribed? If both strands were copied, they would produce two different RNA molecules, since they are complementary, not identical. Each RNA would code for a different protein, leading to conflicting gene expression. Complementary RNAs would bind to each other, forming double-stranded RNA which cannot be translated into protein, would render transcription ineffective. 5.5.1 Transcription unit. Definition. A transcription unit is a segment of DNA that is transcribed into RNA and contains promoter start site, structural gene, RNA coding region, terminator stop site, strands involved, strand type polarity role, template strand 3', 5' used by RNA polymerase to build RNA, coding strand 5', 3' same as RNA sequence, except T replaced by U. Even though the template strand is used to make RNA, the strand that looks like the RNA coding strand is used as reference for gene location. Let's decode how an RNA sequence is written from a DNA template strand. Step 1. Identify the DNA template strand. Given in 3' to 5' direction. 3', ATG, 5'. Step 2. Write the RNA sequence by complementing the bases. Remember, pairs with U, G pairs with A, G pairs with C, C pairs with G. The RNA sequence will be 5' versus 3'. Step 3. Bonus check the coding strand. The coding strand is the same as the RNA sequence, except T is replaced with U. 5'. This is 3'. So RNA matches this, with U instead of T. Final RNA sequence. 5' versus 3'. Promoter and terminator. Component location, WRT coding strand. Function. Promoter upstream, 5' end, RNA polymerase binding site and start point. Terminator downstream, 3' end, signals RNA polymerase to stop transcription. Note: switching promoter and terminator reverses the coding/template strand roles. RNA is synthesized from the template strand in 5' to 3' direction. Quick recap. Feature transcription DNA replication. Strand used one strand template, both strands. Base pairing AU, GCAT GC. Final product RNA DNA. Enzyme RNA polymerase DNA polymerase. Whole DNA copied. No, only genes. Yes, entire genome. Coding strand used. No, used only as a reference. Both strands copied. 5.5.2 Transcription unit and the gene. What is a gene? A gene is the functional unit of inheritance, located on DNA, responsible for coding RNA or proteins. However, defining a gene precisely in terms of DNA sequence can be complex because genes can code for mRNA, coding for proteins, tRNA, transfer RNA, or rRNA, ribosomal RNA. And regulatory elements may be involved too. Cistron concept. Term meaning. Cistron, a segment of DNA that codes for one polypeptide. Monocistronic, found in eukaryotes, has one gene per transcription unit. Polycistronic, found in prokaryotes, has multiple genes per transcription unit. Gene structure in eukaryotes. Most eukaryotic genes are split genes, meaning component description. Exons expressed sequences appear in mature RNA. Introns intervening sequences do not appear in mature RNA. During RNA processing, introns are removed and exons are spliced together to form the final RNA. Important note: this split gene arrangement, exons plus introns, is not found in prokaryotes, where genes are usually continuous, no introns. Regulatory sequences. Even though regulatory regions, like promoters or enhancers, do not code for RNA or protein, they control when and how much gene is expressed, influence inheritance and expression of traits. These regulatory sequences are sometimes called regulatory genes, though technically they are non-coding DNA regions. Quick summary chart. Feature eukaryotes, prokaryotes. Gene structure split, exons plus introns. Continuous, no introns. Type of gene monocistronic, polycistronic. Regulatory sequences present. Yes. Processing required. Yes, splicing of RNA. No, direct translation possible. Example: human genes, E. coli operons. 5.5.3. Types of RNA and the process of transcription. 3 major types of RNA, in all living cells. RNA type, full form function. mRNA messenger RNA carries genetic code from DNA to ribosome, template for protein. tRNA transfer RNA brings specific amino acids to ribosome and decodes mRNA codons. rRNA ribosomal RNA structural and catalytic role in ribosome, site of protein synthesis. Transcription in bacteria, prokaryotes. All 3 RNAs are transcribed by a single RNA polymerase. RNA polymerase is DNA dependent and works in 5'3" direction. It binds to the promoter to start transcription. Nucleoside triphosphates, NTPs, are used as substrates. It opens the DNA helix and adds complementary RNA bases. Only a short segment of RNA remains attached to the DNA at a time. Steps of transcription in prokaryotes. Step description. Initiation: RNA polymerase plus sigma factor binds to promoter and starts RNA synthesis. Elongation: RNA polymerase adds RNA nucleotides using DNA template. Termination: RNA polymerase plus Rho factor detaches at terminator site. RNA is released. Key insight: RNA polymerase alone can only elongate RNA. It requires sigma for initiation and Rho for termination. Coupled transcription and translation in bacteria. No nucleus equals no separation between transcription and translation. Translation can begin while mRNA is still being transcribed. Transcription in eukaryotes. 3 different RNA polymerases in nucleus. RNA polymerase function. RNA pol transcribes rRNA, 28S, 18S, 5.8S. RNA pol 2 transcribes pre-mRNA HNRNA. RNA pol 3 transcribes tRNA, 5S rRNA and RNA. Post-transcriptional processing in eukaryotes. The initial RNA formed equals hnRNA, heterogeneous nuclear RNA. It is non-functional and must be processed before translation. 1. Splicing. Removes introns, non-coding. Joins exons, coding sequences. 2. Capping. Adds methylguanosine triphosphate G to 5' end. 3. Tailing. Adds 200 to 300 adenine A nucleotides at 3' end. This is called the poly-A tail. Final processed RNA equals mRNA, which exits the nucleus to undergo translation. Why so complex in eukaryotes? Suggests ancient genome organization. Introns may represent remnants from the RNA world. The splicing process shows the regulatory role of RNA in evolution. Let's compare transcription between bacteria, prokaryotes, and eukaryotes, summarized in a quick table. RNA polymerase types. Bacteria. Only one RNA polymerase does all the work. Eukaryotes. They have 3 types. RNA polymerase I for rRNA. RNA polymerase II for mRNA. RNA polymerase III for tRNA. Post-transcriptional steps. Bacteria, none. The RNA is ready as soon as it's made. Eukaryotes, yes. They undergo splicing, capping, and tailing before RNA is functional. Transcription-translation link. Bacteria coupled. Transcription and translation happen simultaneously. Eukaryotes separate. Due to the nuclear membrane, transcription happens inside the nucleus, translation in the cytoplasm. hnRNA. Heterogeneous nuclear RNA. Bacteria absent. Eukaryotes present. This is the precursor form of mRNA. Introns in genes. Bacteria rare or absent. Eukaryotes present. Their genes have introns, non-coding regions, and exons. Conclusion. Prokaryotes keep it simple. Eukaryotes have more steps, more complexity, but also more regulation and control. 5.6. Genetic code. Insert summary plus notes. What is the genetic code? It's the set of rules by which information in mRNA is translated into a sequence of amino acids, protein. Unlike DNA replication or transcription, translation involves converting a nucleotide language into a protein language, amino acids. No direct complementarity exists between bases and amino acids, but experimental evidence showed changes in DNA lead to changes in proteins. Historical contributions. Scientist contribution. George Gamow proposed codon must be a triplet to code for 20 amino acids. 4 superscript 3 equals 64 possible codons. Har Gobind Khorana developed method to chemically synthesize defined RNA sequences. Marshall Nirenberg created cell-free system to study protein synthesis. Severo Ochoa used polynucleotide phosphorylase to synthesize RNA without a DNA template. Salient features of genetic code. Feature description. Triplet code 3 nucleotides equals 1 codon equals 1 amino acid. Total 64 codons. Degeneracy. Many amino acids have more than one codon, e.g. phenylalanine equals UUC. No punctuation. Codons are read continuously, without skipping. Universal. Same codon means same amino acid across organisms, except mitochondria, some protozoa. Start codon August. Codes for methionine, met, and signals start of translation. Stop codons UAA, UAG, UGA. Do not code for any amino acid. Decoding the mRNA sequence. Given mRNA sequence. August UUC. Now break it into codons and match each with amino acids. Codon amino acid. August methionine met. UUU phenylalanine Phe. UUC phenylalanine Phe. UUC phenylalanine Phe. UUU phenylalanine Phe. UUU phenylalanine Phe. UUC phenylalanine Phe. Given mRNA sequence. August UUUC. Let's decode this using the genetic code chart. August codes for methionine, the start codon. UUU and UUC both code for phenylalanine. So, the amino acid sequence is methionine, phenylalanine, 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 phenylalanine. That's how the genetic code translates mRNA into a chain of amino acids, the first step in building proteins. 5.6.1 Mutations and genetic code. Insert summary. What is a mutation? A mutation is a sudden heritable change in the DNA sequence. Mutation can be large, deletion/rearrangement of gene segments, or small, change in a single base. Types of small mutations. Point mutations. Type effect example. Substitution. One base is replaced by another. Sickle cell anemia. Glue-val. Insertion. One or more bases are added. Alters codon reading frame. Deletion. One or more bases are removed. Alters codon reading frame. Sickle cell anemia. A classic point mutation. A single base substitution in the gene for beta-globin causes glutamate, GLU, to be replaced by valine, VAL. This leads to production of abnormal hemoglobin, causing sickle-shaped RBCs. Understanding frame-shift mutations. Analogy with English words. Genetic code is read in triplets, like words of 3 letters. Original. RAM has red cap. Codon-like triplet reading. RAMHASREDCAP. Insertion of one letter e.g. B. Between has and red. Read it like RAM has BREDCAP. Reading frame is disturbed. Different words. Codons. Insertion of 2 letters, e.g. BI. RAM has BIREDCAP. Even more distortion of codon pattern. See change in meaning of words in both sentences. Insertion of 3 letters, e.g. big. RAM has big red cap. Reading frame remains intact. Only one new codon added. Conclusion. Frame shift versus codon insertion. Mutation type. Reading frame protein product. Insert/delete 1 or 2 bases shifted. Leads to incorrect codons and premature stop codons. Severe effect. Insert/delete 3 bases, or multiple of 3. Not shifted. Just extra or missing amino acids. Mild or tolerable effect. Key terms recap. Point mutation. A change in a single base pair. Frame shift mutation. Caused by insertion/deletion of 1 to 2 bases. Shifts codon reading frame. Codon. A triplet of nucleotides coding for 1 amino acid. Triplet code. Reading pattern of 3 bases at a time. 5.6.2 tRNA. The adapter molecule. Insert summary. Concept origin. Crick's adapter hypothesis. Francis Crick proposed that a special adapter molecule is needed to read the genetic code on mRNA, carry specific amino acids to the ribosome for protein synthesis, This adapter was later identified as tRNA, transfer RNA, originally known as sRNA, soluble RNA. What is tRNA? A small RNA molecule, 70-90 nucleotides long. It has two functional sites. Anticodon loop. Reads the mRNA codon by base pairing. Amino acid acceptor end. Binds to a specific amino acid. Structure of tRNA. Secondary structure, cloverleaf model. Has the following important features. Anticodon loop. Triplet of bases complementary to mRNA codon. Recognizes and binds to mRNA codon during translation. Amino acid acceptor arm. End with sequence CCA at 3' end. Binds to specific amino acid. CC loop. Contains modified bases. Helps in ribosome recognition. HU loop. Helps identify correct aminoacyl tRNA synthetase enzyme. Tertiary structure. tRNA folds further to form a compact 3D shape, like an inverted L. Specificity of tRNA. Feature details. Amino acid specificity. Each tRNA binds to only one specific amino acid. Anticodon specificity. Each tRNA recognizes a specific codon on mRNA. Initiator tRNA. Special tRNA, with anticodon for August, starts translation. No tRNA for stop codon's termination is handled by release factors, not tRNA. tRNA's role in translation. Links the language of nucleotides, codons, to the language of proteins, amino acids. Acts as a bridge between mRNA codon and the correct amino acid. Key takeaways. Term description. tRNA transfer RNA. Reads codons and brings amino acids. Anticodon triplet on tRNA complementary to mRNA codon. Amino acid arm site where amino acid attaches, at 3' end. Initiator tRNA. Special tRNA that starts translation with methionine. No tRNA for stop codon's release factors stop translation instead. 5.7. Translation, protein synthesis. Insert Class 12 Biology. Molecular basis of inheritance. Definition. Translation is the process of synthesizing a polypeptide, protein, from the mRNA sequence. It involves reading the nucleotide triplets, codons, on mRNA and linking corresponding amino acids together to form a polypeptide chain. Key concepts. Concept details. Small blue diamond template mRNA provides the codon sequence. Factory ribosome, site of protein synthesis. Workers tRNA brings amino acids to ribosome. Bond-formed peptide bond between amino acids. Energy used ATP, used during tRNA charging/amino acylation. Catalyst ribosome, specifically 23S rRNA in bacteria, acts as ribozyme. Process of translation. The entire translation process can be divided into 3 major steps. 1. Initiation. Small ribosomal subunit binds to mRNA at start codon, August. The initiator tRNA carrying methionine, August, binds to the start codon. Large ribosomal subunit joins to form the complete ribosome. The ribosome has 2 sites. P site, peptidyl site. A site, aminoacyl site. Elongation. Charged tRNAs bring amino acids to the A site by matching their anticodon with codons on mRNA. Peptide bond forms between amino acids in P site and A site. Ribosome shifts to next codon called translocation. The previous tRNA exits, and the next charged tRNA enters. Process continues, amino acids added one by one. Termination. When a stop codon UAG is reached, no tRNA binds. Instead, a release factor binds, causing the polypeptide chain to detach. Ribosome subunits disassemble. Translational unit. The region of mRNA between start and stop codon that codes for polypeptide. UTRs, untranslated regions. 5' UTR and 3' UTR are non-coding regions on mRNA. Help in regulation and efficiency of translation. Charging of tRNA, amino acylation. Each tRNA is linked to its specific amino acid using ATP. Enzyme. Aminoacyl tRNA synthetase. Charge tRNA equals tRNA plus amino acid. Term meaning. Codon 3 base sequence on mRNA coding for an amino acid. Anticodon 3 base sequence on tRNA complementary to mRNA codon. tRNA transfer RNA. Brings specific amino acid to ribosome. Polypeptide chain of amino acids. Peptide bond between two amino acids. Amino acylation charging tRNA with amino acid. Ribozyme RNA molecule with catalytic activity. 23S rRNA. Initiator codon August. Codes for methionine. Release factor protein that binds to stop codon to end translation. 5.8. Regulation of gene expression. What is gene expression? Gene expression refers to the process by which genetic information, DNA, is used to synthesize proteins, or functional RNAs. Not all genes are active at all times. Regulation ensures that the right proteins are made at the right time and in the right amount. Levels of gene regulation in eukaryotes. Level what's regulated. 1. Transcriptional control of mRNA formation, i.e., when and how much mRNA is made. 2. Post-transcriptional regulation of RNA splicing and processing. 3. Transport control over movement of mRNA from nucleus to cytoplasm. 4. Translational control over initiation or efficiency of translation into protein. Example. Enzyme regulation in prokaryotes E. coli. Enzyme, beta-galactosidase, breaks lactose into glucose plus galactose. Condition. If lactose is present, enzyme is needed, gene is expressed. If lactose is absent, enzyme is not needed, gene is repressed. Conclusion. Gene expression is regulated by the environment/metabolic state to save energy and resources. Gene regulation in prokaryotes. Main regulation happens at transcription initiation level. RNA polymerase needs help to bind the promoter region. Key regulatory elements. Element role. Promoter region where RNA polymerase binds to initiate transcription. Operator sequence adjacent to promoter where regulatory proteins bind. Repressor protein binds to operator to block RNA polymerase. Activator protein helps RNA polymerase to bind and initiate transcription. The operon model, prokaryotes like E. coli. A functional unit of gene expression that includes structural genes, code for proteins. Operator, regulatory DNA sequence. Promoter, binding site for RNA polymerase. Regulatory gene, produces repressor. Key example, lac operon. Component function. Lac operator DNA sequence bound by lac repressor. Lac repressor regulatory protein that binds operator and blocks transcription. Inducer, lactose. Inactivates the repressor, turning on gene expression. Only lactose, inducer, can inactivate the lac repressor and allow gene expression. Summary points. Gene expression can be regulated at multiple levels. Transcription to translation. Environmental/metabolic signals influence whether a gene is on or off. Operons are common in prokaryotes for regulating related genes together. Regulation helps in efficient use of resources and cellular adaptation. Lac operon. Gene regulation in prokaryotes E. coli. What is an operon? An operon is a functional unit of DNA in prokaryotes that includes structural genes, regulatory sequences, promoter operator, controlled by a single regulatory mechanism. Why lac operon? E. coli bacteria prefer glucose as an energy source. When glucose is absent and lactose is present, E. coli activates the lac operon to break down lactose into glucose plus galactose using the enzyme beta-galactosidase. Components of lac operon. Part description. Structural genes encode proteins for lactose metabolism. Lax produces beta-galactosidase, breaks lactose. Lacey produces permease, helps lactose enter the cell. LACA produces transacetylase, function not clear. Promoter P binding site for RNA polymerase. Operator O binding site for repressor protein. Regulatory gene Lacey produces repressor protein, constantly expressed. How it works. Inducible system. 1. In absence of lactose, off state. Repressor protein from Lacey binds to the operator. This blocks RNA polymerase. No transcription of structural genes. No enzyme to digest lactose. 2. In presence of lactose on state. Lactose acts as an inducer. It binds to the repressor, changing its shape. Repressor cannot bind to operator. RNA polymerase transcribes lax, Lacey, LACA. Enzymes are produced to metabolize lactose. Summary table. Condition repressor bound. Genes transcribed. Enzymes produced. No lactose yes no no. Lactose present no inactivated, yes yes. Important terms. Term meaning. Inducible operon turned on in presence of a substance, like lactose. Repressor protein that blocks gene expression. Inducer molecule, like lactose, that disables the repressor. Polycistronic mRNA-1 mRNA codes for multiple proteins. Lax, Lacey, LACA. Key points for exams. Lac operon is a classic inducible operon model. Repressor binds operator in absence of lactose. Lactose removes the repressor by acting as an inducer. All three structural genes are transcribed together. Lac operon. Negative regulation. Insert Class 12 Biology. What is negative regulation? Negative regulation is when a repressor protein binds to the operator region of DNA and prevents transcription of structural genes. In the lac operon, negative regulation ensures that enzymes for lactose metabolism are produced only when needed, i.e., only when lactose is present and glucose is absent. Negative regulation of lac operon, no lactose. Condition: lactose is absent. The regulatory gene, LAC-I, constantly produces a repressor protein. This repressor binds to the operator region O. It blocks RNA polymerase from transcribing the structural genes, LAC-Z, LAC-Y, LAC-A. No mRNA is made, and no enzyme is synthesized. Lactose digestion enzymes are not produced, because they're not needed. This is called negative regulation, because the repressor negatively controls gene expression. Lac operon turns on lactose present. Condition: lactose is present. Lactose acts as an inducer. It binds to the repressor, changing its shape. The repressor cannot bind to the operator anymore. RNA polymerase can bind to the promoter and transcribe the structural genes. Enzymes beta-galactosidase, permease, transacetylase are synthesized. Lactose is digested and used as energy. Summary table. Negative regulation in lac operon. Let's decode how the lac operon works under different conditions. When lactose is absent, the repressor binds to the operator. Transcription is blocked. No mRNA is made. No enzymes are produced. This is an example of negative regulation. The system is OFF. When lactose is present, the repressor is inactivated by lactose. It does not bind to the operator. Transcription starts, mRNA is produced. Lac operon enzymes are synthesized. This is called induced expression. The system is on. In simple words, no lactose, operon off. Lactose present, operon on. Important notes. Negative regulation helps save energy by switching OFF unnecessary genes. Lacey gene is constitutive, always on, to make the repressor. Lac operon is an example of an inducible system controlled by negative regulation. 5.9 Human Genome Project, HGP. What is the human genome? The human genome is the entire DNA sequence in a human, which includes all the genes and non-coding regions of DNA in our chromosomes. Why was the HGP launched? DNA contains all genetic information. To understand how DNA sequence leads to traits or diseases. To compare differences between individuals based on DNA. Launched in 1990, completed in 2003. Called a mega project due to its scale, data, and cost. Magnitude of the project. Human genome is almost equal to 3 billion base pairs, BP. Estimated cost per base. $3. Total is almost equal to $9 billion. If printed, would need 3,300 books of 1,000 pages, each page with 1,000 letters. Required supercomputers and bioinformatics tools. Main goals of HGP. Identify 20,000 to 25,000 genes in human DNA. Determine the complete 3 billion base pair sequence. Store the data in digital databases. Improve tools for data analysis. Transfer new technologies to industry, e.g. medical, biotech. Address ethical, legal, and social issues, ELSI. Global collaboration. Coordinated by U.S. Department of Energy, National Institutes of Health (NIH). Major contributors. Wellcome Trust UK, Japan, France, Germany, China, and others. Completed in 2003, after 13 years. Importance of the project. Disease understanding and treatment. DNA variations lead to disease clues. Personalized medicine. Future potential to prevent and treat genetic disorders. Non-human genomes. Model organisms sequenced for comparison and applications. Bacteria. Yeast. C. elegans worm. Drosophila fruit fly. Plants like rice and Arabidopsis. What is bioinformatics? A branch of biology combining computers, software, and biology to analyze large biological data. Especially useful in HGP for storing, retrieving, and analyzing DNA sequences. Exam highlights. Topic key fact. Start year 1990. Completion year 2003. Human genome size 3 billion base pairs. Estimated genes 20,000 to 25,000. Main tools use DNA sequencing, cloning, computers. Key field developed bioinformatics. Ethical issues covered ESI. Major model organisms used yeast, Drosophila C. elegans rice. Methodologies used in Human Genome Project. The Human Genome Project HGP used two major strategies to decode the entire DNA sequence of humans. 1. Expressed sequence tags ESTs. Gene-focused approach. Goal. Identify only the genes that are actively expressed as RNA. Focus was on coding regions, exons. Helped quickly identify functional parts of the genome. Think of it like locating active light bulbs in a building by seeing which ones glow. 2. Sequence annotation. Whole genome shotgun approach. Goal. Sequence the entire genome, including both coding genes, non-coding, introns, intergenic, DNA. After sequencing, assign functions to different regions, annotation. Called a blind approach because everything was sequenced, not just known genes. Key steps in genome sequencing. Step description. 1. DNA isolation. Total DNA is extracted from human cells. 2. DNA fragmentation. DNA is cut into smaller fragments because full-length DNA is too long for sequencing machines. 3. Cloning fragments are inserted into suitable vectors and cloned in host cells. 4. Amplification. DNA is amplified, copied, to ensure there's enough material for sequencing. 5. Sequencing fragments are sequenced using automated DNA sequencers based on Sanger's method. 6. Alignment. DNA fragments are aligned based on overlapping regions to reconstruct the entire sequence. 7. Annotation. Assign functions to specific sequences. Identify genes, regulatory regions, etc. Hosts and vectors used. Type description. Hosts bacteria and yeast. Vectors BACs, bacterial artificial chromosomes. And YACs, yeast artificial chromosomes. Purpose used for cloning DNA fragments. Role of computers and software. Due to huge data, humans could not align or analyze sequences manually. Bioinformatics tools and computers were used for fragment alignment, sequence assembly, data storage and annotation. Final challenges. Last chromosome, chromosome 1, completed in May 2006. Mapping genetic features like restriction site polymorphisms, microsatellites, repetitive DNA. These were crucial for creating physical and genetic maps. Exam highlights table. Feature description. Two main approaches: ESTs, coding focus, sequence annotation, whole genome. Cloning vectors used Bax and Waces. Sequencing method Sanger method, automated. Key person Frederick Sanger. Software role overlap alignment and annotation. Last chromosome sequenced chromosome 1 in 2006. Genetic map tools microsatellites. Restriction site polymorphisms. 5.9.1 Salient features of human genome. HGP highlights. Feature description. 1. Genome size. Human genome has 3,164.7 million base pairs, BP. 2. Average gene size. A typical gene is 3,000 bases long, but can vary widely. 3. Largest human gene. Dystrophin gene, about 2.4 million bases long. 4. Total genes identified 30,000 genes, much lower than earlier predictions of 80,000. 1. 40,000. 5. Genetic similarity. 99.9% of DNA sequences are identical in all humans. 6. Unknown gene functions. More than 50% of identified genes have no known function yet. 7. Protein coding. DNA. Less than 2% of the genome codes for proteins. 8. Repeated sequences make up a very large portion of the genome. Not coding but important for chromosome structure, dynamics, evolution. 9. Most and fewest genes. Chromosome 1 has the most genes, 2,968. Y chromosome has the fewest, 231. 10. SNPs, single nucleotide polymorphisms. About 1.4 million SNPs found, pronounced SNPs. Useful in disease gene mapping, small blue diamond tracing human ancestry and history, understanding repeated sequences. These are stretches of DNA that repeat many times, hundreds or thousands. Although they don't code for proteins, they help understand chromosome behavior, provide clues on evolution and structure, are used in DNA fingerprinting. Why these findings are important. Challenge earlier assumptions, e.g., fewer genes than expected. Help in personalized medicine, disease diagnostics, and evolutionary studies. Pave the way for genome editing, gene therapy, and biotechnology. For exam quick revision. Question answer. How many base pairs are in the human genome? 3.16 billion. 3,164.7 million BP. Largest gene in the genome. Dystrophin, 2.4 million bases. Number of protein coding genes. Around 30,000. Percent of DNA coding for proteins. Less than 2%. Similarity between human genomes. 99.9% identical. What are SNPs? Single base changes in DNA. Help trace disease and ancestry. 5.9.2 Applications and future challenges. Applications of Human Genome Project, HGP. Field potential use/impact. Biomedical research enables systematic study of all genes instead of one at a time. Personalized medicine, tailored treatment based on a person's unique genetic makeup. Molecular diagnostics, early detection and precise diagnosis of genetic disorders. Understanding complex diseases, studying how multiple genes interact in diseases like cancer, diabetes, etc. Functional genomics, study of all transcripts, mRNA, and their expression in specific cells/tissues. Proteomics and systems biology, understanding how thousands of proteins and genes function in networks. Agricultural biotechnology, identifying genes for drought resistance, yield improvement, pest resistance, etc. Evolutionary biology, comparative genomics, helps understand evolutionary relationship. Future challenges. Challenge description. 1. Data interpretation, huge volume of sequence data, but deriving meaningful biological insights is complex. 2. Cross-disciplinary expertise requires collaboration between biologists, computer scientists, chemists, physicists, and engineers. 3. Ethical, legal, and social issues, ELSI. Concerns over privacy, genetic discrimination, and misuse of genetic data. 4. Technology development, need for faster, cheaper, more accurate sequencing and analysis tools. Key shift in research approach. Then, study of one gene at a time. Now, genome-wide, high-throughput, system-level analysis of all genes, all transcripts in a tissue, all protein interactions. Exam quick recap. QA. What is the main shift in research due to HGP? From gene-by-gene studies to whole genome, systems biology approach. What does HGP help understand better? Gene functions, gene expression, disease mechanisms, evolution, cellular networks. One major future challenge. Making sense of massive sequence data and applying it effectively. 5.10 DNA fingerprinting. What is DNA fingerprinting? DNA fingerprinting is a technique to identify individuals based on their unique DNA sequence patterns. It targets specific regions of DNA that show high variation, polymorphism, among individuals. Why is it possible? Because 99.9% of DNA is the same in all humans. Only 0.1% variation, 3 million base pairs, is responsible for individual differences. This variation is mostly found in non-coding repetitive DNA regions, which are used for fingerprinting. Repetitive DNA and satellite DNA. Type description. Repetitive DNA, short DNA sequences repeated many times. Satellite DNA, repetitive DNA separated during centrifugation as distinct peaks. Types, mini satellites like VNTRs, microsatellites, etc. Role, show polymorphism, useful for identification, paternity tests, and forensics. DNA polymorphism. Polymorphism equals genetic variation among individuals, caused by mutations, germline or somatic. If a variation is found in greater than 1% of the population, it's called a DNA polymorphism. Mostly occurs in non-coding regions. Mutations here don't affect reproduction and accumulate over generations. What is VNTR? VNTR equals variable number of tandem repeats. Short sequences repeated in tandem. Number of repeats varies among individuals. Forms the basis of fingerprinting as it generates unique patterns. Steps in DNA fingerprinting. Traditional method by Alec Jeffreys. Step process. 1. Isolation of DNA from the sample, blood, hair, semen, etc. 2. Digestion with restriction enzymes to cut DNA into fragments. 3. Gel electrophoresis to separate DNA fragments by size. 4. Blotting, transfer of DNA fragments to a nylon or nitrocellulose membrane. 5. Hybridization using radio-labeled VNTR probe. 6. Detection using autoradiography, shows bands unique to individual. Each person, except identical twins, shows a unique banding pattern, a DNA fingerprint. Modern method uses PCR, polymerase chain reaction, instead of Southern blot. Requires only 1 cell worth of DNA. Faster, more sensitive, and non-radioactive methods now common. Applications of DNA fingerprinting. Application purpose. Forensic science, criminal identification, rape cases, murder investigations. Paternity testing, resolve disputes about biological parentage. Population genetics, study of genetic diversity and evolution. Gene mapping helps in human genome analysis. Wildlife conservation, identify individuals in endangered species. Track illegal poaching. Important terms recap. Term definition. Satellite DNA non-coding DNA with repeated sequences. VNTR mini-satellite DNA with variable repeat numbers. Polymorphism heritable variation in DNA sequence. Autoradiography detection of radio-labeled DNA using X-ray film. Quick exam flash facts. 99.9% of DNA is same in all humans. Only 0.1% causes variation. DNA fingerprinting uses non-coding, highly polymorphic sequences. Used in forensics, paternity, and gene mapping. Alec Jeffreys invented the VNTR-based DNA fingerprinting technique. Here are the detailed answers to your Insert Biology textbook questions from Chapter 6. Molecular basis of inheritance, Class 12. 1. Group the following as nitrogenous bases and nucleosides. Nitrogenous bases nucleosides. Adenine cytidine. Thymine guanosine. Uracil. Cytosine. Nitrogenous bases. Adenine, thymine, cytosine, uracil. Nucleosides equals nitrogenous base plus sugar, e.g. cytidine equals cytosine plus ribose. 2. If a double-stranded DNA has 20% cytosine, calculate the percent of adenine. C equals 20%, G equals 20%. Total equals 40%, C plus G. Remaining 60% equals A plus T. Since A equals T, A equals 30%. Answer: 30% adenine. 3. Complementary DNA strand. 5', 3'. Direction. Given strand. 5' ATG CATG 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 C. 3' end. Complementary strand. Reverse the base pairing direction. 5' G CATG 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 CAT-3'. Answer: 5'. G CATG 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 CAT 3'. 4. Sequence of mRNA from coding strand. Coding strand equals 5' ATG CATG 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 C-3'. In transcription, mRNA is same as coding strand, but with U instead of T. mRNA equals 5'. AUG CAUG 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 C-3'. 5. Which property led Watson and Crick to propose semi-conservative replication? Complementary base pairing of the two DNA strands led Watson and Crick to propose that during replication, each strand serves as a template, producing one parental and one new strand. Complementarity and base pairing suggested semi-conservative mode of replication. 6. Types of nucleic acid polymerases. Template synthesized enzyme. Template DNA, synthesized DNA. Enzyme DNA-dependent DNA polymerase. Template DNA, synthesized RNA. Enzyme DNA-dependent RNA polymerase. Template RNA, synthesized RNA. Enzyme RNA-dependent RNA polymerase. Template RNA, synthesized DNA. Enzyme RNA-dependent DNA polymerase. Reverse transcriptase. 7. How did Hershey and Chase differentiate DNA and protein? They used radioactive isotopes. Superscript 3 superscript 2p to label DNA. Phosphorus is in DNA, not protein. Superscript 3s to label protein. Sulfur is in protein, not DNA. After infection of E. coli with labeled phage, only superscript 3 superscript 2p entered the bacterial cell, proving that DNA is the genetic material. Differentiate between A. Repetitive DNA versus satellite DNA. Repetitive DNA, satellite DNA. DNA with repeated sequences, highly repetitive DNA that forms distinct peaks in centrifugation. Includes micro/mini satellites, a type of repetitive DNA. B. mRNA versus tRNA. mRNA-tRNA. Carries genetic code from DNA, brings amino acids to ribosome. Linear structure, cloverleaf structure. C. Template strand versus coding strand. Template strand, coding strand. Used for RNA synthesis, has the same sequence as mRNA, except TU. 3' 5' direction 5' 3' direction. 9. 2 essential roles of ribosome during translation. Provides site for protein synthesis. Catalyzes peptide bond formation between amino acids. Peptidyl transferase activity. 10. Why does lac operon shut down after some time even with lactose? Once lactose is consumed, it no longer acts as an inducer. Without inducer, the repressor binds again, and lac operon is turned off. 11. Functions. A promoter. Initiation site for RNA polymerase to begin transcription. B. tRNA. Transfers amino acids to ribosomes during translation. C. Exons. Coding sequences in a gene that remain in mature mRNA. 12. Why is the Human Genome Project a megaproject? Because it involved sequencing 3 billion base pairs. Required international collaboration. Huge funding. $3 billion. Took 13 years and advanced bioinformatics and automation. It was one of the largest coordinated scientific efforts in biology. 13. What is DNA fingerprinting? Mention its application. DNA fingerprinting. A technique to identify individuals based on polymorphic repetitive DNA, like VNTRs. Application. Used in forensics, paternity testing, crime investigation, and population studies. 14. Brief descriptions. Transcription. Synthesis of RNA from a DNA template using RNA polymerase. B. Polymorphism. Genetic variation at a particular locus that occurs with frequency greater than 1% in a population. C. Translation. Process of protein synthesis using the mRNA code on ribosomes with the help of tRNA and enzymes. D. Bioinformatics. The application of computational tools to analyze biological data, especially DNA, RNA, and proteins. MCQs. Molecular basis of inheritance. 1. DNA replication is called semi-conservative because DNA is synthesized in short fragments. B. Only one strand of DNA is copied. C. One of the two strands is newly formed, other is parental. D. DNA synthesis is continuous on both strands. Answer Z. 2. Which of the following forms the backbone of DNA? Purines and pyrimidines. B. Ribose and phosphate. C. Sugar and nitrogen base. D. Sugar phosphate. Answer D. 3. Which enzyme unzips the DNA strands during replication? DNA polymerase. B. DNA ligase. C. Helicase. D. Topoisomerase. Answer Z. 4. Which of the following is not a component of a nucleotide? Nitrogen base. B. Pentose sugar. C. Phosphate group. D. Ribosome. Answer D. 5. In eukaryotes, DNA is wrapped around RNA. B. Histone proteins. C. Actin. Ribosomes. Answer B. 6. Which of the following is involved in transcription? DNA polymerase. B. RNA polymerase. C. DNA ligase. D. Peptidyl transferase. Answer B. 7. Which of these statements about genetic code is false? A. It is triplet in nature. B. It is overlapping. C. It is universal. D. It is non-ambiguous. Answer B. A. The start codon is August. B. UA. Udi. Yuga. Answer. 9. VNTRs used in DNA fingerprinting are a type of rRNA. B. Satellite DNA. C. mRNA. D. Histone. Answer B. 10. Which of the following is used to detect hybridized DNA in DNA fingerprinting? X-ray film. B. Autoradiography. C. Centrifuge. D. Microtome. Answer B. 11. Which component of a gene is transcribed but not translated? Intron. B. Exon. C. Promoter. D. Enhancer. Answer. 12. Which of the following human chromosomes has the fewest genes? X. B21. 1. Y. Answer D. 13. The largest known human gene is hemoglobin. B. Insulin. C. Dystrophin. D. Keratin. Answer Z. 14. Hershey and Chase used radioisotopes of A. Carbon and nitrogen. B. Phosphorus and sulfur. C. Calcium and iron. D. Oxygen and hydrogen. Answer B. 15. DNA polymerase adds nucleotides in 5'3" direction. B. 3'5" direction. C. Random direction. D. Circular motion. Answer. HOTS. High-order thinking skills. Questions. 1. Why is DNA fingerprinting considered highly reliable even with minute DNA samples? Answer. Because of the high degree of polymorphism in VNTR regions, and the sensitivity of PCR, polymerase chain reaction, which can amplify DNA even from a single cell. 2. If one strand of DNA has the sequence 5' GUCTAG 3, write the mRNA sequence transcribed from this region. Answer. First write the template strand. Template equals 3'. CA5. mRNA equals 5'. G3. 3. How does the presence of repetitive DNA aid in evolution and speciation? Answer. Repetitive DNA undergoes frequent mutations without affecting phenotype, since they are non-coding, leading to genetic variability that is essential for evolution and speciation. 4. Why is only one strand of DNA transcribed during transcription? Answer. Because simultaneous transcription of both strands would result in two complementary mRNA molecules that code for different proteins, leading to functional chaos in cells. 5. How does the lac operon act as a model for gene regulation? Answer. It demonstrates inducible gene regulation, where the presence of lactose removes the repressor and allows transcription, highlighting control mechanisms in prokaryotic gene expression. 6. Why does the genetic code show degeneracy? What is its evolutionary significance? Answer. Degeneracy, multiple codons for one amino acid, minimizes the impact of mutations, making the genetic code more error-resistant, aiding survival and evolution. 7. What could be the consequence if introns are not removed during mRNA processing? Answer. Translation would result in a faulty or non-functional protein, since introns do not code for amino acids and may introduce nonsense sequences.